All right, guys, this notes packet covers actually topics 2.3 through 2.5. Now, if you've noticed the numbering, this is AP specific. I did not create these numbers, but essentially, this is Unit 2, Topic 4. And so it might feel a little bit weird that we are skipping topic 2.3. I think it best fits kind of towards the end of this notes packet as opposed to the beginning. And so even though if you kind of put it in sequential order according to College Board, it would come first, we will see 2.3 at the end. Bear with me, trust me in that I think it will make more sense if we push it to the end of this notes packet. So we're going to jump into topic 2.4, which is essentially the membrane structure, a cell membrane's structure. Something new that you'll notice at the top of this notes packet, I went ahead and pasted in what is called an enduring understanding for this unit. Now, I did not make this, again, taken straight from College Board's curriculum for this class. An enduring understanding is something that essentially, by the end of this unit, something that is big enough and important enough and kind of a big enough topic that you will remember it years and years and years and years from now. So, the enduring understanding that we're going to be exploring over the next couple of topics is this: cells have membranes that allow them to establish and maintain internal environments that are different than their external environments. So, the next couple of this notes packet and the one following, we're really focused on cell membranes and how they maintain stable environments in an ever changing external environment or world. So let's start with always the structure. Let's start with the membrane structure. So this picture hopefully maybe looks a little bit familiar to you. This is our friend, the phospholipid. So back from macromolecules, remember phospholipids are a type of lipid. They are an amphipathic lipid, which means, vocab refresher, it means that it's going to have both polar and nonpolar regions. So the head of the phospholipid, which is made up of a bunch of phosphates, and you can see it here in this yellow circle, this is going to be polar. And the tails are the fatty acid tails. So these are non-polar, big hydrocarbon tails. So the tails can be either saturated or unsaturated. So remember, saturateds are the straight-tailed, unsaturateds are bents. And we'll come back to why tails would be saturated or unsaturated, or possibly a mix of both within a cell membrane. There is a functional difference. And so we'll come back to that. Keep in mind anything that is polar, like the head, is going to be hydrophilic, and anything that is nonpolar, like the tail, is going to be hydrophobic, which explains what happens when you put lipids into water. So think about, you have a head that is polar and tails that are nonpolar. If you dropped this into water with other phospholipids just like it, the heads would sort of orient themselves on the outside. They kind of all clump together and form a sphere. The heads would orient themselves to the outside solution, and the tails would kind of cluster themselves here in the middle, trying to get away from the water because they are nonpolar. So the phospholipid bilayer, that means bilayer means two layers. It means that there's two layers of phospholipids within the cell membrane. And when they orient, they're always going to look like this. When they get into position, they're always going to look like this. Their heads will be on the outsides, their tails will be tucked in the middle, trying to get away from any kind of watery environment. If we look next at a protein, there are a lot of proteins wedged in the membrane. And so if we look at these pictures here, you can see this sort of blue protein and a blue protein here, and then this big thick middle region of the blue protein. And then you can see these little purple components. So the key tells us something. The key tells us that within this, if we'll use this as an example, within this overall protein, the parts that are in blue, that part of the protein is going to be made of amino acids that are hydrophobic, which means that they're nonpolar. And then the sort of fuchsia parts of the protein are going to be made of amino acids that are polar, which should maybe resemble to you, hopefully, the structure of the overall phospholipid bilayer, meaning look at all these nonpolar tails here in the middle. That's where the nonpolar part of the protein is located. And look at the polar heads. That's where the polar component of the protein is located. Here's a different kind of protein. It doesn't go all the way through the membrane, but you find that same trend. Wherever the nonpolar fats are, that part of the protein wedged in the membrane is also going to be nonpolar. Wherever the polar heads are, the part of the protein that's hanging out there will mimic the polarity of the phosphate heads. And so that's kind of an interesting thing about proteins. Membrane proteins, are also amphipathic. They will mirror the polarity of the area in the protein to the area in of the membrane. So if the protein is going to go through the membrane, it's going to be nonpolar in the middle to mirror these tails of the phospholipids that are nonpolar, and then the outer components are going to be actually polar, mirroring the phospholipid heads. Same thing here is a very complex protein. It's very big. The outer portions that are hanging out with the polar phospholipid heads will be polar as well. And then the inner portion of the protein right here that's hanging out in the middle of the cell membrane with the the nonpolar lipid tails, this part will also be nonpolar to mirror those tails. So there's two main categories of proteins. There are integral proteins. Those are proteins that go all the way through the membrane, and as such, they are sometimes referred to as transmembrane proteins because they transect the membrane. Now, these are really good for moving things from the outside to the inside or vice versa of the cell because they actually create kind of a tunnel or a channel to move things through. So we'll come back to integral when we talk about movement a little bit more. The other kind of protein is called a peripheral protein. So think about, you've probably heard of your peripheral vision. That's the vision that's on the side of your eyes, sort of. And so these are proteins that are attached to either the inside or the outside of the membrane. They could be attached to another integral protein themselves, or they could be attached directly to a lipid. So, two main categories, and I could give you an example. This right here is a peripheral protein. This very well could be a peripheral protein attached to a part of an integral protein. This gives us a lovely picture of lots of different ways that proteins might look in a diagram. So, these right here, alpha helix curls, remember those in our protein folding? So this right here is a very simple integral or transmembrane protein. These are more complex. So this one weaves itself multiple times through the membrane, but it still goes all the way through the membrane. This one right here goes all the way through, creates a big giant kind of channel or hole that things can move in and out of the cell. This one, even though it doesn't look like, it doesn't go all the way through the membrane, it's embedded pretty intensely, and so it is still considered an integral membrane protein. Here are some peripheral proteins. So the peripheral parts are what are in teal. So here's a peripheral protein that is attached to an integral protein. Here is another peripheral protein that is attached to an integral protein. This one is on the extracellular world, which means it's on the outside of the cell. This one was on the inside of the cell. Here's a peripheral protein that's just attached to a lipid. And then over here are specific, what are called lipid linked protein membrane proteins, we don't talk about those as much, but essentially they are linked directly to a phospholipid. If we look at our next component, cholesterol is going to be found in the membrane. So cholesterol right here are remember, a ringed fat. And so they are, shouldn't surprise you, hanging out with the other fats, which are the tails of the phospholipids primarily. And they're kind of wedged in between the overall phospholipids. So what do? They're a structural component of the cell membrane. They help keep the membrane held together when temperatures start to change. Cholesterol is only found in animal cell membranes, so it's not in plants or protists or fungus or even prokaryotes. And again, what specifically are they doing? We'll come back to cholesterol in class this week, but essentially it's keeping the membrane nice and fluid as temperatures change. As it gets really cold or as it gets really hot, cholesterol can work in both situations to maintain a nice, fluid, flexible, stable membrane. The last component that I want to kind of toss out there are glycoproteins and carbs. So, this right here, could maybe put this in the world of a carbohydrate. Remember, glycogen is a big, giant structural carb, or I'm sorry, energy carb. And then glycosidic linkage are the kinds of bonds that are formed between carbohydrates. So, that prefix glyco could help you to associate to, hey, we're going to stick a whole bunch of sugars onto either a lipid and create something called a glycolipid, or we're going to stick a whole bunch of sugars onto a protein in the membrane and call it a glycoprotein. So when we attach carbs to a phospholipid, we get a glycolipid, or a protein and we get a glycoprotein. So what do these do? Actually, they do a lot of things. One really important thing that glycolipids and glycoproteins do is related to cell recognition. So, your immune system, if you don't know anything about it at all, at least maybe you could hold on to the idea that your immune system is there to kind of protect your body and specifically to help defend against foreign invaders, which means your immune system needs to be able to recognize the cells that belong to your body as your cells, or else it's going to attack them. Your immune system needs to be able to recognize that things like bacteria and virus and fungus is not supposed to be there, and it launches an immune response to try to get rid of those. So, your glycolipids and glycoproteins can function in the immune system as cell recognition, as identifying your cells as belonging to you in the hopes that your immune system doesn't attack your own body. It doesn't always work perfectly, which lays the basis for if you've ever heard of autoimmune diseases, but we'll save that conversation for another day. So, antibodies are a key component of our immune system. These are really important glycoproteins. We also use glycolipids and glycoproteins on the surface of your membrane as receptors for when cells start to talk to each other. So cell communication, which we get into in the next couple of units, will be important to kind of use these glycoproteins and glycolipids. And then they're also going to create some adhesion. If two cells need to stick to each other, they can do that by using these sticky carbs attached to the lipids and the proteins. We're going to dip our toes into membrane function just for a second. I want to talk about something called the fluid mosaic model. So, the fluid mosaic model is the name given to the model that is the cell membrane. So, let's watch this video first, and we'll come back and finish these notes.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
Okay, so fluid here in the name of our model of the membrane, what science kind of proposes the membrane looks like and acts like. The fluid comes from the idea that number one, everything is kind of moving around in the membrane. You saw in the video that the proteins are sort of doing this little dance throughout the membrane. The big green things were sort of swirling around. They're constantly moving. Same thing with these phospholipids. Phospholipids can sort of swap spaces. They never flip flop in the sense that the top flip flops with the bottom phospholipid in the bilayer, but the neighboring side by side phospholipids sort of do these little circular dances around each other. So there's a lot of action, a lot of movement. The fluid also represents the idea that the membrane is flexible, which the video introduced. Now, mosaic, I, I'm betting that you all have probably seen, oh,
Between the
here,
living
you've probably seen a mosaic, right? So in art class, a mosaic is something that you kind of take small pieces and put them together to make an overall larger structure. And so the mosaic term here comes from the idea that the membrane is made up of lots of different little pieces pieced together for an overall function. Lots of proteins and phospholipids and cholesterol, glycoproteins, glycolipids, things that we've just talked about, all put together into this fluid structure. Now, last thing, we're going to address some things that keep the membrane nice and fluid. So one of these things comes from the lecture right here, and the other two, quote, things will come from an article that we're going to read in class together. So this little GIF of the cell membrane, but watch, again, lots of motion, lots of fluidity. You have things moving around the bilayer. This is a lipid raft, which basically is a big hunk of phospholipids that are attached to something, typically a protein, that sort of move together as a collective throughout the membrane. So, things that impact whether or not the membrane is going to be fluid. The first thing is the idea that it is a mosaic. It is made up of a lot of different things. The proteins are moving around, the lipids are exchanging positions, they're flip flopping, or I'm sorry, they're sort of circling each other. The side by side phospholipids are circling each other in almost like a do-si-do action. If you know anything about square dancing, maybe some of you have done square dancing in PE before, but the lipids are moving, and we see the movement of these big giant lipid rafts as well. So because all these things are moving, it is keeping the membrane nice and fluid. Now, the other two factors that are increasing fluidity or maintaining fluidity are the tails in the phospholipids. Remember, I said they could be both saturated or unsaturated. So we're going to see the tails in our article and kind of what's going on there. And then we're going to talk about cholesterol, because cholesterol is the other big thing that impacts fluidity. But again, those two things come from an article that I have for you. So you are done with this particular section of notes.